Okay, welcome back, Bio 1. We're continuing on with, this is Chapter 12, Section 3, RNA and Protein Synthesis Part 2. Today we're going to take a look at the process known as transcription, and this is going to tell us how RNA is made using DNA. So the first step is, what is transcription? And it says RNA molecules are produced by copying part of a nucleotide sequence of DNA. So that's just saying taking part of the building blocks of DNA and they make it into a complementary sequence in RNA. And this process is called transcription. So all that means is that it's taking a part of DNA to make RNA is all that it does. So RNA, it's making DNA, it's making RNA using DNA. This process is called transcription. And then this requires the enzyme known as RNA polymerase. So we use DNA polymerase in DNA replication. So we're using RNA polymerase in transcription. So during transcription, RNA polymerase will bind to DNA and it will separate the DNA strands. Remember, that's a DNA double helix. So the first step is RNA polymerase comes in and it will separate our DNA into separate strands. And then RNA polymerase will just use one strand of DNA as a template. So a template is just kind of something to work off of, from which the nucleotides are assembled into a strand of RNA. So it's just taking one strand and using that one strand as our guide on how to make RNA. And then this can't just happen in any area. RNA polymerase will bind only to regions of DNA known as promoters. So promoter is going to promote the binding of RNA polymerase, so that might be an easy way to remember that. The promoters are an area where RNA polymerase will bind, and these are signals in DNA that indicate to the enzyme where to bind to make RNA. So that's pretty much what I just said, that it's only going to bind in that certain area, and that's just kind of the signal to the RNA polymerase. And what that kind of looks like here, our big brownish, goldish glob there is binding to the DNA, and that's kind of our signal to start building our nice new RNA with uracil instead of thymine now. And so some RNA editing, what kind of going along with this process is saying that the DNA of eukaryotic genes contains sequence of nucleotides that are called introns, and these are not involved in the coding for proteins. So this is kind of almost the opposite of what you think. If you think introns, that would almost mean inside, that it's going to be included, but they are not involved in coding for proteins. However, our DNA sequences that will code for proteins are called exons. So again, almost sounds kind of opposite, but you need to keep those two straight, that exons are going to be involved for our coding. And when RNA molecules are formed, introns and exons are copied from our DNA. And just kind of taking a look at this more specifically, again, our introns are cut out of our RNA molecules, whereas the exons are then spliced, and splice is just kind of a fancy term to almost be glued together to form mRNA. So that's just some pretty, just kind of know that the difference, that introns are not going to be used to make our RNA molecules, where exons will. Again, that kind of sounds backwards, but just kind of going to have to memorize that fact that exons are used and introns are not. So kind of going along with our next step here, and kind of looking at RNA, is our genetic code. So our genetic code is the language of mRNA instructions. So it's kind of how our mRNA will know what to do. So our genetic code is written using 4 letters, which are 4 bases, A, U, C, and G. Remember, adenine, uracil, cytosine, and guanine. So those are going to be used, again, they're for RNA, that's why we have a U instead of a T, and that's going to kind of be how our cells kind of know what to do and what to make, what type of proteins to make, things like that. And being some more specifics about the genetic code, is they consist of things known as codons. Remember, codon is part of the genetic code, and it will consist of 3 consecutive nucleotides on mRNA. So nucleotides, again, our building blocks are A, our U, and our G on mRNA that specify a particular amino acid. And amino acids are basically the building blocks of proteins. So in order for us to make proteins, we need amino acids. In order for us to make amino acids, we need codons. So these codons will specify what amino acids to make. And here's and then again, each codon specifies a particular amino acid that's to be placed on a polypeptide chain. Polypeptide chain is just a fancy way to say protein. And again, I just said that. And then some amino acids can be specified by more than one codon. So what that means is we have an AUG and we have AAC. These could, in theory, make one amino acid. You don't have to have just one amino acid for every codon. That's not how it works. So we could have multiple make up the same one. Here's a picture of what the genetic code looks like. I will give you a picture that I think is easier to read in the notes. It might make a little more sense than just this one right here. And then just to finish off this video, there is only one codon, AUG, that can either specify the amino acid methionine or it will serve as a start codon for protein synthesis. So it can serve one or two functions, the main one being it will tell us, hey, go ahead and start making these proteins. Then there are 3 stop codons that do not code for any other amino acid, and these stop codons signify the end of a polypeptide. And again, polypeptide, remember, is a fancy word for a protein. Remember that for the quiz, it will be on there. That polypeptide is another word for protein. So that's it for this video. Let me know if you have any questions.